A very good morning to all the delegates. Myself, Dr. Shilpa Harak, is here to begin with the second session. For our second session, we have the revered Dr. Pooja Chawla ma'am. She is a professor at the University Institute of Pharmaceutical Sciences and Research, Baba Farid University of Health Sciences, Faridkot, Punjab. Looking at her educational background, ma'am has a M.Pharm and PhD in pharmaceutical chemistry. She has 23 years of experience in research, discovery, and development of biologically active pharmaceuticals. Looking at everything we do is measured in the terms of outcomes, and that comes for an academician or a researcher has publications and achievements. Just a glance at ma'am's publication shows that she has published 9 books, 40 book chapters, and more than 200 papers in national and international journals. She is also reviewer for international journals of repute. She is a guest editor of 2 reputed journals. First, CNS and Neurological Disorders Drug Targets with an impact factor of 2.8 and Current Organic Chemistry with an impact factor of 2.5. Both are published by Bentham Sciences. She is also appointed as an academic editor of PLOS One journal, which is quite a reputed one, with an impact factor of 3.2, and associate editor in Health Sciences Review Elsewhere publication. She has guided till date 68 postgraduate projects, and her research team includes 4 research scholars who are pursuing their doctorate degree. Her research interest is mostly based on design and synthesis of various heterocyclic moieties, including 4-thiazolidine diones, 2,4-thiazolidine dione, coumarin, pyrazolines, Schiff-base derivatives, to name a very few. All of these are targeted as medicinally important agents. I once again welcome Dr. Pooja and request her now to kindly take over the session. Please, ma'am.
Thank you so much, Dr. Smita Shelke, for such a nice introduction. First of all, I would like to thank Principal Sir Dr. Sunil Amrutkar for giving me such an opportunity and for organizing such a beautiful series of webinars, which are definitely fruitful for all the pharmaceutical, or I should say the medical fraternity, be the students, be the teachers, be the researchers, or the other learning members. I would also like to thank Vice Principal Dr. Prashant Pingale. I am very happy to share that I have met with the Dr. Prashant in an online mode only, where I was hosting a program, and today Dr. Prashant has invited me, and I'm really thankful to him. I'm really thankful to the webinar coordinator, Dr. Smita Shelke, and the entire IQAC, as well as all the management team of Dr. M.S. Busavi College of Pharmaceutical Education and Research, Nasik. It's really a big privilege for me to be with you all and interact with you all and the students online. I think it's a national webinar, so many students across the nation are watching this, and I am hopeful that I'll be able to guide them or just make them learn anything. And today only one more thing I have learned, that is how to work on this new platform, and really thankful. And you are so patient that to make me learn about the streaming on the StreamYard. Thank you so much. So I'll be going to my presentation. Just acknowledge me whether it is visible.
Yes, it is visible.
Okay.
And working.
So as ma'am
I will
has
be in the backstage now, ma'am.
Pardon, ma'am?
I will be in the backstage. I will be in the backstage now.
Okay. So the topic of my presentation today is the therapeutic journey of 4-thiazolidinones as a promising scaffold. I would like to inform you or share with you that this 4-thiazolidinone moiety This particular scaffold, it is very dear to me, and I consider it to be my third child, as I have worked a lot and I'm still learning to work on this particular pharmacophore or scaffold, as you say. Coming to the introduction for 4th thiazolidinones, these, as we all know, that heterocyclic chemistry, the all the medicinal chemistry or the organic chemistry, we are nothing without heterocyclic chemistry. So it's a classical subject of immense practical and theoretical importance, is an inexhaustible resource of novel compounds, small rings hydrocycles containing nitrogen, sulfur, and oxygen. They have been under investigation for a long time owing to their important medicinal properties. There are numerous biologically active molecules whose framework includes five-membered ring containing true heteroatoms. As I have already mentioned that heterocyclic chemistry, there are five-membered, four-membered, six-membered or fused rings. But today, as my topic is 4th thiazolidinones, so I am focusing on the five-membered ring containing two heteroatoms. So among this is the 4th thiazolidinone ring, which is a core structure in various synthetic pharmaceuticals. As the name says, it is 1,3-thiazolidine, 4 ons. They are the heterocyclic nuclei that have an atom of sulfur and nitrogen at position 1 and 3 respectively, and a carbonyl group at position 4. They have been subjected to an extensive study in the recent years. The 4th thiazolidinone scaffold, it is versatile and has featured a number of clinically used drugs. They exhibit a broad spectrum of biological activity like antimicrobial, antihistaminic, anti-inflammatory, anticonvulsant, antitubercular, and anti-HIV, to name a few. Coming to the chemistry of 4th thiazolidinones, these are the derivatives of, as you can see, thiazolidine with the carbonyl group in position 4, as shown in figure 1. The numbering starts from S. As a rule of IUPAC system, the substituents in the position 2, 3 and 5, they may be varied, but the greatest difference in structure and properties is exerted by the group attached to carbon atom in position 2, that is R and R dash, as in case of B, or X, as in case of C. So such groups include alkyl or aryl in case of B, oxygen in case of C, where it comes out to be the new molecule or scaffold, that is 2,4-thiazolidine diones. Sulfur in case of rhodonine, which is also a very active molecule. Imino, that is pseudo-thiohydantoin, in which aryl or alkyl groups, they replace hydrogen. They are named as derivatives of 2-imino 4-thiazolidinones and hydrazine. That is, X is this moiety and named as 4-oxo 2-thiazole 2-hydrazones of aldehydes or ketone. Variations in substituents attached to nitrogen atom and methylene carbon are possible for structures represented by B and C. Here you can see. So their parent structure, it may be modified varying substituents at positions 2, 3 and 5. These are three positions which are available where any modification can be carried out, giving the nucleus different physiological and biological properties. The carbonyl group at position 4, it is highly unreactive, because if this 4 position carbonyl is replaced by any other group, then the entirety or integrity of the 4th-thiazolidinone, as the name indicates, it will be gone. The 3 unsubstituted 4th-thiazolidinones, they are usually found to be solids and decompose on melting. They do not go sharp melting point, they will be decomposing. The position 3, that is N-alkyl substituted thiazolidinones, they possess lower melting points, and the 4th-thiazolidinones possessing no alkyl or aryl substituents, they show some solubility in water. As you all, being the chemistry students, you all will agree with me, as the substitutions increase or else the bulkiness increases, it will ultimately lead to the decrease in the solubility in water. So coming to the molecular spectra, because we all know that chemistry or the organic chemistry or the synthetic chemistry without analytical or the molecular spectral characterization, it is nothing. So what has been seen in general, as coming to the ultraviolet spectroscopy, as I already tell my students that ultraviolet spectroscopy, it gives just a bleak idea about what sort of conjugation or the conjugated double bond system or the conjugation or the unsaturation which is present in the molecule. Only that can be observed or that can be identified. So in case of rhodonine derivatives, where there is sulfur at position 2, So here you can see it has been found that they give two characteristic peaks in the case of rhodonine derivatives at 250 and 290 nanometer or millimicron due to CN bond in conjugation with the thione group and to the dithio group. In case of B, the thione and the amide groups. And a slight bathochromic shift, it has been observed on decreasing the strain due to 5-membered ring of 4-thiazolidinones by an increase of the ring size to 6-membered or by the ring opening. In case of, as far as the infrared spectroscopy is concerned, that we all know that infrared spectroscopy tells about the functional group, the major functional group which are present in case of the organic molecule. So here we can see that as there is a carbonyl group, so a strong and a characteristic peak for 4-thiazolidinones and a twin peak in case of 2,4-thiazolidine is observed between 1760 and 1655 centimeter inverse due to carbonyl stretching, and it is the most significant peak that while you are carrying out IR spectroscopy, you are not in a position to neglect that particular peak for a carbonyl group, highly intense, highly sharp peak or the band. 4-thiazolidinones possess near peak in the region of 3100 to 3400 centimeter inverse, thus revealing about the NH stretching in the 3 position when the nitrogen is having a hydrogen, that is, it is not in the unsaturated position. Now coming to the synthesis of these 4-thiazolidinone nuclei, the literature is replete. You can see number of methods for the synthesis. More often than not, it is a 3-component reaction involving an amine, a carbonyl compound, and a mercapto acid. And the process can be either a 1-pot 3-component condensation or a 2-step process. Caesar et al. And Vicini et al., they have carried out a lot of research on this, and they have reported another method of synthesis of 4-thiazolidinones by use of thiocyanate, alkyl thiocyanate, or ammonium thiocyanate with hydrazide or acetamide, followed by the treatment with ethyl bromoacetic acetate or the sodium acetate in the Newengel condensation at the 5th position, that I will tell in my forthcoming slides. Otana et al., they have reported synthesis of 4-thiazolidinones using starting material that is N-propyl, N-phenyl thiourea working under acidic conditions. And there are various methods or the papers that have been published by Otana et al. Or various other scientists across the globe. Coming to the microwave assisted synthesis, that is 3-component regioselective 1-pot cyclo condensation for a series of novel spiroindole thiazolidinones or 5-thyrylidine 2-amino 4-thiazolidinone have also been reported, because as while we are talking about the synthesis or the organic chemistry or the synthetic chemistry, one are the conventional methods and the other is the non-conventional method. And you all will agree with me that nowadays, due to the environmental hazards or the pollution, the green chemistry has come to a boom. And it is really the need of the hour today that the scientists are shifting towards the green chemistry. Now coming to the conventional method, as I have already mentioned in my previous slide, there is the synthetic route, it involves an amine, a carbonyl group, and a mercapto acid. And the main synthetic routes to 1,3-thiazolidine 4-ones, it involves 3 components, that is an amine, carbonyl, and a mercapto acid. The classical method can be either a 1-pot synthesis or it may be a 2-step process. The reaction here, you can see it begins with the formation of an imine, that is nitrogen of amine attacks the carbonyl sorry, of the aldehyde or a ketone, which undergoes attack by generated sulfur nucleophile followed by intramolecular cyclization on elimination of water. Here you can see in this thing, there is a reaction between the aldehyde and amine. With loss of water, it will lead to the imine, which is also called as the Schiff base, which on a reaction with mercaptoacetic acid or mercapto acid, it will lead to the ring cyclization. Coming to the microwave assisted synthesis, it can lead to a 1-step synthesis. Here I have taken the article from the literature. The authors have used benzene, but as we agree that benzene is carcinogenic, so you can replace benzene with any other solvent which is not harmful. And here, what is the beauty of the microwave assisted synthesis or the ultrasonication reactions or the non-conventional method, here you can see the reaction time. It can reduce from hours or days to just few minutes. Okay, and the reaction conditions are not that drastic, and we don't have to keep waiting for the hours or the days. And it has also been observed, and we have also carried out some reactions. The yield is improved. You can get as high as 90% yield. So this is the probable mechanism of reaction. How it carries out, as I have already mentioned, in the first step, it is the synthesis of with the loss of water. It is a synthesis of Schiff base or the imine, where the in the second step, the mercapto group, it attacks at this position, and ultimately there is a ring closure, and you get the fourth thiazolidinone ring system. Coming to the physical properties, like N3 substituted thiazolidinones, they are usually solids. Their substitution at N3 position and often melt with decomposition, but sometimes substitution at this position lowers the melting points, leading to some oily compounds which are really difficult to recover. Now, thiazolidinones that do not carry N-aryl or higher alkyl substitutions, they are partially soluble in water owing to their bulkier structure. And some low molecular weight 4-thiazolidinones, they can be recrystallized from water. The introduction, as we all know, the more you introduce the substituents, the decrease in the solubility in water takes place. And they, whether substituted or non-substituted, have a characteristic intense odor, and introduction of substituents decrease the intensity of odor. Thiazolidinones, they are usually colorless or possess some characteristic color. I would like to share with you that when I was carrying out my M.Pharm, in the second year, there is lot of labor. There is lot of masti also, because अकेला काम करने से कुछ नहीं होता जब तक आप enjoy नहीं करते. And the supervisors, sometimes they have a tendency. Now I am the supervisor, so we have the tendency to just raid the lab whether the students are working or not. And when my supervisor, my guide sir, when he used to enter my lab, he on the door only he used to understand that yes, the synthesis is going on, because a peculiar smell of the sulfur, it takes place when the ring formation is there. Coming to the reactions of the 4-thiazolidinones, they undergo limited reactions or transformations. As you can see here, on the fifth position, it is highly reactive, and they possess the nucleophilic activity and can attack an electrophilic center. The reaction occurs in the presence of a base and an anion of 4-thiazolidinone attacking species. And here we can see it is the best suitable site for the Navangel condensation to take place. The fourth position carbonyl group, it is highly unreactive, but in some cases, like in case of Lucent's reagent, which is used for the thiation, you can see this carbonyl group is replaced by, this oxygen is replaced by the sulfur. And oxidation of 2,3 position can also take place in some of the conditions under the oxidative reaction conditions. Coming to those stereochemistry, theoretically, in case of 2,3 disubstituted molecule where 2 and 3 are occupied, the nitrogen is having occupation or the substitution. 2 diastereomers 1 and 2, they are possible as shown in the figure below. Vigorita scientists, they have done conformational studies, and they have seen that in case of this structure, the preferred configuration is the one position, that is in which C2, this one, C2 proton and one of the methylene protons are in cis 1,3 di-equatorial relationship, because of the fact that the phenyl group prefers axial position to avoid steric hindrance, which may be present due to pyridine ring, which is present on the nitrogen at position 3. Now coming to the biological activities of 4-thiazolidinones, here you can see it has got a plethora of activities, starting from antimicrobial, anti-inflammatory, analgesic, antioxidant, anti-ulcer, antidiabetic, you name it, you get it. But the thing is, it is only the game of the substitutions that can take place only those 3 particular positions, that is position 2, 3 and 5. Now I will switch over to the research that is carried out by myself as well as my group, our group. The first reaction, the first slide that I'm showing you here right now, it is the work that was carried out by me, and a total of 35 derivatives, they were synthesized, and they were evaluated for antimicrobial, anti-inflammatory, analgesic, or you can call it as an antinociceptive, as well as the antioxidant activities. Here you can see the primary requirement is the primary amine group. Here you can see these are the substituted anilines. Here I have taken the case of three chloro position. A total of 5 series were synthesized: chloro, 3 and 4 dichloro, 3 chloro, 4 fluoro. So, number of substitutions, 5 series were synthesized. The first step involves the chloroacetylation of the amino group. This one, primary amine group. It can take place depending upon the weather. It can take place at the room temperature also. But here we have to maintain the temperature below from 0 to 5 degrees centigrade and completely moisture free, because chloroacetyl chloride, it is highly reactive, it is highly irritating, and once you carry out the reaction at with this particular moiety, that is chloroacetyl chloride, you have itching, irritation all over the skin. Wherever you touch with your hands, you will have itching. So being a chemistry teacher, I would like to inform all the students that we have to take the particular precautionary measures, because you must have heard one saying that "Savdhani hati durghatna ghati." So chemistry is nothing but chemical mystery. जितना ये कॉपी में और इस लैपटॉप पे अच्छा लग रहा है रिएक्शन, it is entirely different whenever you enter a lab. So this chloroacetylation reaction, it is very carefully if you carry out, this is beautiful, and this two position, this is called as the chloroacetamide. This chloroacetamide, in the second step, it undergoes heterocyclization in the presence of ammonium thiocyanate, in the presence of alcohol as a solvent, and it can take from 4 to 5 to 6 to 9 hours also. Simply you can reflux it, or you can keep on stirring or refluxing with stirring. And in the final step involves, you see here we have carried out the synthesis. The substitutions at 2 position is already occupied. We are not touching the 3rd position. We have touched the 5th position. Here active methylene group, what it will do, it will undergo Navanjal condensation with the help of, in the presence of various aldehydes, substituted aldehydes, acetic acid and sodium acetate refluxing. So here you can see what we intended to synthesize. It was 2,5 di-substituted compounds, and these compounds were subjected for initially we carried out antimicrobial activity. After that, we proceeded for anti-inflammatory and analgesic and antioxidant. So what was the conclusion for this particular time? That 35 derivatives were synthesized, 5 series, and each series contained 7 substitutions with respect to the aldehydes in the 3rd position. So they were subjected to antimicrobial, anti-inflammatory, anti-nociceptive, and antioxidant activities using couplet, red paw edema model, tail flick method, and DPPH assay method respectively. None of the compounds was found to produce toxicity at a dose of 50 mg. That was the dose that I selected for my work. LD50 of the most active compound of the each series, it was found to be more than 500 mg per kg. So 50 mg was selected. Most of the compounds, they showed significant anti-inflammatory activity and analgesic activity. Also showed good activity against the chosen bacteria. We chose 2 gram positive group, 2 gram negative, and 1 fungal strain. Gram negative positive, Staphylococcus aureus and Bacillus subtilis, Pseudomonas aeruginosa and E. coli as the gram negative bacteria, and Candida albicans for the fungal strain. And it was found that the electron withdrawing groups like chloro, nitro on the arylidine ring at the position 5, that enhance the activity. Chloro group at para. See, first of all, which groups affected the activity and the position of those groups, like chloro at 4th position, ya para position, and nitro at the meta position showed the maximum activity, whereas electron donating groups like hydroxy and methoxy decreased the anti-inflammatory activity. Most of the compounds exhibited significant anti-nociceptive activity or analgesic activity, but there was not much affected by the electron withdrawing or releasing groups. They were influenced by the activity was influenced by the arylidine moiety present on the 5 position that I have already mentioned. The presence of an electron withdrawing group in the arylidine nucleus produced potent antimicrobial activity. In the B series, where I took 3-chloro-4-fluoro. The nitro group at the meta position of the arylidine ring demonstrated the most potent antimicrobial activity. I can share the papers also, that publications which I got from this work. The SAR studies revealed that the presence of fluoro group at the phenyl imino group, that is in the first step, at the 2 position, had marked influence on the antibacterial activity, and 4-fluoro group, in addition to 3-chloro, they were more active against chosen bacteria. When I chose only 2-fluoro, it was good activity. Only 3-chloro, it was also good activity. But when I chose 3-chloro, 4-fluoro aniline, it was taken, then it shows the best antimicrobial activity against the chosen strains. And in case of anti-inflammatory activity or analgesic activity, most of the compounds exhibited promising results. In case of A series, where I took the 3-chloro derivative, they showed the maximum anti-inflammatory activity. Compounds had chloro at para and nitro at the meta position. These are the 3 publications that are from this particular work in the medicinal chemistry research by Springer publisher, and a part of the work was presented in the FIP conference which was held in Hyderabad in 2011. Now coming to the next work, here you can see again, as I told, primary amino is the requirement for this reaction to be carried out. In this, the aryl hydrazides were taken as the source of primary amine, followed by the formation of Schiff base. And here you can see the activity of the substitution at the N3 position was carried out using mercaptoacetic acid, which I showed in that synthesis slide. Here you can see the synthesis was carried out at 2, substitutions were carried out at 2, 3 as well as the 5 position, and a total of 14 derivatives were synthesized, and they showed promising anti-inflammatory as well as the antioxidant potential of these compounds. And this paper was has been published in the Current Organic Chemistry by Bentham publishers. In our next work, a total of 12 derivatives were synthesized. Here again, substituted anilines were taken. Here the solvent system, you can see maintaining 0 to 5 degrees centigrade, and stirring for just 1 to 2 hours, it was reduced. And in the presence of dichloromethane, the chloroacetylation was carried out, followed by heterocyclization, and the last step involved the Novengel condensation, glacial acetic acid and sodium acetate की presence में. Here a total of 12 derivatives were synthesized, and it was, you can see, we did not use the traditional aryl aldehydes. Here what we used were we used the quinoline aldehyde, quinoline moiety was taken, indole moiety was taken, and furan moiety was taken in addition to the typical phenyl ring, that is the aryl aldehydes. So in this case, the compounds were evaluated for anti-cancer activity, anti-breast cancer activity against the MCF7 anti-cancer, the breast cancer cell lines. And it was found that at R2, here you can see in the arylidyl fifth position, the quinoline showed better results than indole, which in turn was more than chlorinated phenyl, and the furan was among the series, it was the least active. And replacing quinoline and indole motifs with furan rings decrease the anti-breast cancer activity. Coming to the substitution at R1 position, here this one, you can see electron withdrawing groups like 3-chloro or 3-chloro-4-fluoro enhanced anti-cancer activity. And the order of the activity was 3-chloro more than 3-chloro-4-fluoro, more than 4-methoxy, more than trimethoxy. Here, in order to establish a particular structure activity relationship, what we used the electron withdrawing groups as well as electron donating groups. And what happens if we take more than one electron donating group or withdrawing group? So for that case, we took 3-chloro, 3-chloro-4-fluoro, methoxy or trimethoxy derivatives. The in silico studies were carried out, and it was found that the thiazolidine and the pyridine hybrids, they interact with the amino acids like phenylalanine and glutamine in target protein binding sites, influencing their activity as well as the selectivity. And this paper was published in Polycyclic Aromatic Compounds with impact factor of 2.4. In our another work, another student, what she carried out, Archana, total of 10 derivatives synthesized, anti-cancer activity was carried out, the method is established. See, you can see the variation from this slide to this slide. The reaction conditions are different, and the reaction times can also be different depending upon the conditions. And I also would like to mention that the reaction, the climatic conditions also have an effect on our synthesis. Once you carry out reaction in the month of May, June, in the month of December or September, the environmental conditions, the temperature, the humidity, they all affect your reaction. That's why I always tell my students, my research team, that whatever you cannot see, that if your seniors have carried out this work in 2 hours or 3 hours, you will also carry out that work in that much time only, because you cannot predict the environmental conditions and the manual handling also. So this paper was published again in the polycyclic aromatic compounds, and here SCR studies told about that electron donating groups at aryllidine group in position 5 increase the antioxidant activity and decrease by the presence of electron withdrawing groups. Chloro substituted phenyl imino group at second position enhance the anti-inflammatory. Here we carried out anti-inflammatory, anti-cancer activities also. So these moieties improved the anti-inflammatory as well as the anti-cancer activity. Uh, on this slide, I will come a bit later. This is the recent work last year only. It has been published in Journal of Molecular Structure with impact factor of 4. Here we carried out the synthetic part. Here you can see we have taken different substituted pyridine moieties. We have tried some more also. Um, Here you can see 10 derivatives were synthesized, and they could fit very well into the pockets of a COX-2, and electron donating groups on the phenyl group linked to the thiazolidinone ring enhance the anti-inflammatory activity, whereas electron withdrawing groups like nitro or chloro, they reduced the activity, likely due to the decrease in the electron density affecting the interaction with the COX-2 enzyme. 3 and 4 pyridine derivatives that we have taken here, they enhance the COX-2 selectivity, possibly due to the better accommodation in the COX-2 binding pockets, and the electron-rich substituents like methoxy or methyl at the para position, they enhance the COX-2 inhibitory activity by improving the hydrophobic interaction. And this paper, here we have carried out the in vitro, in vivo, in silico studies, and MD studies indicated that the compounds were quite stable, and they up to 100 nanoseconds we had got very good interactions. Uh, Another work that was carried out by my student Jakseer, in the paper was published in Letters in Organic Chemistry. We carried out the anti-inflammatory activity as well as the antioxidant activity using DPPH method. And you see why these compounds are so active as anti-inflammatory or COX-2 inhibitors, because here you can see whenever we talk about the COXIBs, you can see the coccyx have a this COX-2 pocket is different from the COX-1 pocket, and only the bulkiest groups can enter the COX-2, not the COX-1. And these coccyx, they have got a butterfly type structure with two wings. Here you can see this iminoaryl group as well as the arylidine group, they act as the wings for the butterfly core, that is the 4-thiazolidinone core moiety. So these compounds, they definitely have the potential to fit into the COX-2 pocket and give the better results. Now I will come to this slide, and two papers have already been accepted, but I cannot show them right now because they are still in the process. We have explored and now we are very much into it, the non-conventional method. Here we have used the ultrasound assisted solvent free synthesis, because whenever we talk about green chemistry, we talk about the solvents, the wastage of the solvents, the pollution, the water pollution, the environmental pollution. So here we have taken, in the first step you can see the Schiff base is formed within 30 minutes after stirring and using sodium sulfate as the and DCM. And the second step involves ultrasonication, absolutely solvent-free synthesis in the presence of mercapto acid. And in the table you can see here, we have synthesized 10 derivatives, and we have not stopped here. The process is going on. Here, 2 and 3 positions, they are they are substituted, and in the yield column, you can see we got up to 97% of the yield. So we have got a good scope in case of green chemistry. This such a beautiful, this wonder nucleus, it can be, a lot of derivatives can be synthesized using conventional as well as the non-conventional methods. And this paper was published in the Journal of Sulfur Chemistry. Now, there are many other papers also, but I would not like to discuss here, because there is one set scheme and a number of derivatives that can be synthesized. But again, it depends upon you what sort of the initiating the starting material you take, what are the conditions. So a lot of work can be carried out on this particular, this wonder moiety, that is called as fourth hyaluronidinone nucleus. Now I will discuss something about the structure activity relationship studies of fourth hyaluronidinones carried out by some other authors. As I have already mentioned, that we have this nucleus has got a number of activities, starting from antimicrobial, anti-diabetic, anti-cancer, and so on. So here you can see in this particular slide that being the organic chemistry person, we have also noticed, or you must have noticed, that the effect of substitution, effect of saturation is also there. So here you can see saturated IAZ displayed 5 to 12 fold. This is the work by other authors only. 5 to 12 fold greater binding affinity than the unsaturated compounds, and the S isomer of the unsaturated IAZ showed better binding affinity to PTP1B as compared to R isomers. In the second part, you can see the benzothiazole moiety, this one, resulted in lower polar surface area, improved Kekoto permeability, and ortho. Not only, as I have already mentioned, not only the derivatives, their position on the particular ring also affects the activity, whether they are causing some steric hindrance, whether the moiety is able to bind with the particular receptor, and for how long that can be bound with that particular receptor. That is also very important. In the next slide, you can see the anti-cancer potential. If a student thinks about that, what sort of activity I can carry out, what sort of substitution I can take for my moiety to synthesize, or what sort of pharmacophore I should take, that solely depend upon your interest, your area of work, or your interest of in that particular area, or the choice of your guide also. That how can you, and one has to think continuously about, if डॉक्टर एक्स कैन डू इट, वाई कांट आई. और कभी कभी मन में ये भी आता कि उसने सोचा होगा, तो क्या अगर मैं ये चीज सोच रहा हूँ, वेदर इट इज पॉसिबल और नॉट. सो लॉट ऑफ देयर कम्स द इंपॉर्टेंस ऑफ लिटरेचर सर्वे. So while choosing, I know many students are listening to me, be the M.Pharm or the PhD scholars or the persons who want to pursue their PhD, be very specific while choosing your moiety. And the more you do the literature survey, the more intense you do the literature survey, you carry out the literature survey, the lesser the hurdles that you will carry out during your research journey. So here you can see in the first part, that is compound inhibit hormone independent DMB induced mammary carcinomas in rats. Here you can see the nitrosubstituent in benzothiazole moiety increase the inhibitory effects. So the list in never ending, as you can see here. This slide shows the anti-hepatitis agents. One may think that why should I think about the anti-cancer or only anti-inflammatory or only this activity. This is purely your choice. You can explore the molecules, and I should say the chemistry, the entire organic chemistry, the entire chemistry, it is the ocean of the active molecules that can be present over there. You only have to dive deeper and deeper and deeper, and you can have the pearls of pharmacophores, which may prove to be active for discovery of various drugs or the new molecules. The anti-inflammatory and analgesic effects, here I have not mentioned my papers or my work because I have already discussed about them in my previous slides. Antimicrobial, 4-thiazolidinones, thiazolidinediones, thiazoles, diazoles, these heterocyclic moieties, you can move forward to start with. If there are very new students or B.Pharm students who just think whether we should carry out we should go for the further studies. You can go for antimicrobial. It really is very interesting and nowadays you understand that as per the IAC guidelines, it's very difficult to take the approval for against the cruelty against the animals. So one can go for the in vitro activities also. These are some of the references that I have included for preparing this presentation. So that's all from my side. I would be happy if you have any questions. And madam has got my contact number, my email ID, my phone number. And in the last, but not the least, because a teacher is nothing without his or her students. So I would like to acknowledge my teachers, my supervisors, my colleagues, and my students who have made me learn day by day, and still today only I am in a learning mode, and I am thankful to my colleagues, my students, the college, the university where I am working today, that is Baba Farid University. I am also thankful to my Vice Chancellor, sir, who has always, Dr. Rajiv Sood, who has always encouraged us for research activities. So thank you so much once again. Thank you.
Yes, thank you, ma'am. We have some questions in the chat. I'll just highlight them and also read it.
Okay.
So here we have a question. In case of thiazolidine diones, which reduction method is best for removing particular double bond after attaching thiazolidine moiety?
See, whenever you have prepared this moiety, it's very difficult to carry out the reduction, and I would not advise to carry out the reduction because it can, destabilize your pharmacophore that you are tending to synthesize. So should not go for the reduction of the double bond because you have synthesized the aryllidine moiety. Na? So first of all, you should not synthesize the 5-aryllidine moiety. If you have synthesized, you should not go for its reduction.
I hope that meets the requirement. We have one more question. I'll highlight it. Okay. What is the activities of 4-thiazolidinedone?
I think Rajendra has not joined earlier. There are ample amount of activities, but if you ask me, the field I am working upon, you can go for its antimicrobial activity. You can go for its anti-inflammatory activity. Anti-tubercular activities are also there, and I would like to mention here, if you want to go for the anti-tubercular activity, you should carry out the substitution at position 3. For carrying out the anti-inflammatory activity, you should go for the substitution at position 2 as well as position 5. And for anti-inflammatory activity, as I have already mentioned in my slide, you should go for the 2 and 5 and take the aryllidine moiety, because as I have already mentioned, that will mimic the butterfly structure of the coccyx, and it will be able to fit into the COX2. It will not be able to enter the COX1. So it will be entirely your COX2 inhibitors, and you will not get any undue effects or side effects of like ulcers or acidity because of the inhibition of the COX-1. They will not be able to enter the COX-1 pocket. Anti-cancer, I forgot to mention. Anti-cancer also. Anti-cancer activity, you can go for that.
Okay, we have one more question by Mr. Rajendra Mahajan only. I think
Yeah.
that also you have answered, but probably he must be seeking something else.
Please, ma'am, I would love to entertain any of his queries. If he can, you can share my contact number with him. No problem at all.
Yes, that will do, ma'am. Let me check if there are any others. Okay. There are very comments on the session was excellent and so.
Thank you. Thank you
So
so much.
yes, I would like to summarize the session. Like actually, thiazolidine ions we know a bit, but today's your lecture was huge, extensive, elaborate, and on not only is the synthetic part, but you also explained about chemistry, physical properties, how to identify like structural elucidation also.
One more thing I would like to mention here, ma'am. Usually, we misspell or we confuse 4-thiazolidinones with thiazolidinediones. They are entirely two
Okay.
different moieties. This is
Yes.
4-thiazolidinone. It is having one carbonyl, and thiazolidinedione. It is having additional carbonyl group at
Carbon.
position 2. Yeah. So
Yes.
they are so both of these moieties, they are very much dear to me. And thiazolidinediones, as you must have heard about glitazones. So they are very good anti-diabetic agents, and they are PPR gamma agonists. And research is going on this particular moiety for thiazolidinones, that people are going towards thiazolidine for anti-diabetic activities.
Yes.
Good
In
precursors for TZD synthesis. There is one question, Ali. You
yes.
can go for that thiourea, and it is so easy. Beta, I don't know whether Sonali is a student or a teacher. One thing I would like to mention here, that thiazolidinedione, it is also available ready made. So what I suggest my students, that you should prepare in your lab, and you can have comparison with the ready made, that it is not that much expensive. So you can go for its synthesis, and you can have a direct comparison like TLC. Chemistry people, ma'am, you will agree with me, we cannot do anything without TLC. TLC tells us about the progress of the reaction. So there are ample amount of substituents that we can go, the starting materials that we can go for it. You can go for one pot synthesis. You can go for multiple step reactions. It is particularly your choice as well as your guide's, preference. Sorry, ma'am, I interrupted you in between.
No, it's all right. We got a question by that time. Okay. So Sonali is saying that she's a SND. She's a faculty somewhere and
Okay,
has not
okay.
yet started work on it.
You can
Okay.
contact me anytime. I have, because ma'am, this was for thiazolidinones only. I have worked a lot on TZDs also. So please share my number with Sonali also. I would love to help her out.
Yes, 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 ma'am. So as I was saying that you have structural elucidation and physical properties, stereochemistry. The part which any medicinal chemist likes is identifying the structure activity relationship. Because once you set the structure activity relationship, then you can think of further derivatives, how to do and what to do,
Yeah.
and also the activity based on it. So that was a quite a wide scope. I'll say about 1/4 thiazolidine fourth thiazolidine diones, which is there. So I like to thank you for the session, and there are number of comments also online on the YouTube that it was an excellent and informative session. So I thank you, ma'am, on behalf of Sir Dr.
Thank
M.S.
you.
Kosabi College of Pharmacy, Principal, Vice Principal, staff, and of course, our management. It is because of speakers like you who are benevolent enough to give us their time and energy and share your research that we are able to conduct such webinars. Thank you once again, ma'am.
Thank you.
We'll go
Thank you so
offline
much,
now.
ma'am. Thank you.
Yes.
Thank you.